everyone. I'm Sidra from Aligarh Muslim University, and my work is on glycoxidated IgG in type 2 diabetes mellitus. Now, glycoxidation is a post-translational modification. Basically, we all know that the modification that occurs in protein as a result of addition of functional group to it is the post-translational modification. It may be enzymatic and non-enzymatic. Post-translational modification alters the physiological behavior of protein, increases functional diversity in the proteome, and can cause pathogenesis. Now, there are various types of post-translational modifications like glycation, nitration, oxidation, carbon amylation, amidation. My work is on glycation and oxidation, together called glycoxidation. So, these post-translational modification has a role in a variety of diseases like diabetes, rheumatoid arthritis, neurodegenerative diseases, endothelium dysfunction, arterial stiffening, amyloid diseases, systemic lupus, erythematosus, microvascular complications, and diabetes and cataractous lens. Now, the two very important post-translational modifications are glycation and oxidation. The two are inextricably interlinked and they mutually enhance each other. Together they form glycoxidation products. The glycation, in short, is the addition of a reducing sugar like glucose to the epsilon amino group of protein to form the glycation products, which first of all early glycation products are formed, followed by the formation of advanced glycation end products. In a period of days to weeks, early glycation end products forms, followed, and in months and years, advanced glycation end products are formed. Now coming to oxidation. Oxidation is basically the attachment, oxidation is when a protein or any other biological macromolecule is damaged by oxygen-free radicals. There are various types of oxygen radicals like peroxide, superoxide, hydroxyl radicals, hydroxyl ion, and my work is on hydroxyl radical. When they damages the protein, they cause cross-linking in the protein. So together glycation and oxidation forms glycoxidation products. In short, glucose forms Schiff base followed by oxidation, and ultimately cross-linked damaged proteins are formed as a result of glycoxidation, which causes chronic inflammation and tissue damage. Proteins modified forms carbonyls, cross-linked products, and various oxidative additives like carboxymethyl lysine, pentosidine, etc. Now, why diabetes? I have taken why diabetes because it is a very widely growing disease with a rate and around 387 million people are worldwide affected by this disease. Now, what happens in diabetes is basically the insulin, pancreas secretes insulin. The insulin receptor is not working in diabetes, as a result of which hyperglycemia occurs. So the uptake of insulin is not proper in diabetes, maybe because of the insulin receptors are not working or there is not proper secretion of insulin from the pancreas. Now, diabetes has a prevalence, very high prevalence in all over the world. Like in Canada, 2.7 million people are suffering from diabetes. USA, Mexico. So it is very highly growing disease. It is a serious concern in India as well. It is a potential epidemic in India, with more than 62% people currently diagnosed with the disease. In 2000, India topped the world with the highest number of diabetes cases, followed by China and US. According to scientist Wilde et al., the prevalence of diabetes is predicted to double globally from 171 million to 366 million, with maximum in India, followed by China and US. There's a very crucial relation in diabetes and glycoxidation. Glycoxidation means glycation followed by oxidation, plays an important role in the pathogenesis of diabetes, and in glycation also free radicals are generated by the auto-oxidation of sugars, lipids in plasma and membrane proteins. So carboxymethyl-lysine, pentosidine are generated by sequential glycoxidation and oxidation. Oxidative stress elevated by continuing cycle of metabolic stress, tissue damage, cell death, leading to increased free radical production. Now, the aim of our study is, because there is a vicious interdependency between glycation, oxidation and diabetes. So we have taken a glycating agent methylglyoxal, a hydroxylating agent, a reactive oxygen species, that is hydroxyl radical, and we have seen the role in diabetes. So the study aims to mimic this situation in vitro. We have worked on a protein IgG and we have modified it with glycating agent and oxidizing agent. Now what we have done is, basically, first of all, we have isolated IgG from the sera. This is the isolated IgG using protein A agarose column, and then we have checked it by UV absorbance spectrometry as well as SDS page. Our results reveal that the isolated protein is IgG, as there is a band of 150 kilodalton that came when the protein was run in page. Now this isolated IgG is glycated by using methylglyoxal as glycating agent, followed by the hydroxyl radical modification of glycated IgG. By hydroxyl radical is generated in our laboratory by Fenton's reaction, in which we have taken FeCl2 and H2O2 in a ratio of 30 micromole. 3:10 ratio we have taken FeCl2 and H2O2. So hydroxyl radical is generated. Now we have modified, ultimately we got hydroxyl radical modified glycated IgG. These are desired protein, the desired modification we got. Now we have done its structural and immunological characterization. Now UV absorbance spectroscopy revealed a very enhanced hyperchromicity in the samples in the IgG protein modified by methylglyoxal as well as hydroxyl radical. It has been found to be 75% hyperchromicity is found in MgOH treated IgG. We have also run two parallel controls to see the only glycation in effect and only hydroxyl radical modification effect. But it has been found that maximum damage to protein occurred when this protein is reacted with methylglyoxal in combination with hydroxyl radical. So pointing a very damaging effect of glycoxidation. It was followed by, we have also done Congo red staining technique to see if the glycoxidated protein, to see the structure of glycoxidated protein. We have done native protein, the Congo red staining samples of native hydroxyl radical modified, methylglyoxal modified and hydroxyl plus methylglyoxal modified. It has been found that in hydroxyl and methylglyoxal modified protein, very high aggregate formation was detected. This shows that there is a high damage. This aggregate may be due to cross links that are formed when the protein is modified by a combination of two. It was followed by the immunological studies. Immunological studies included direct binding and inhibition ELISA, antioxidant parameters like carbonin content estimations and others. So we have collected the sera of 80 diabetes patients and 25 healthy sera was collected. The selection of patients is based on brief medical history, physical examination, and patients with other chronic diseases were excluded from the study. The blood was collected, followed by serum separation, and the sera is stored in aliquots at minus 20 degrees C using 0.1% sodium azide as preservative. These are just the antioxidant parameters which we have done on diabetic sera to see if the sera we have collected, and we found that there is reduced glutathione content in diabetic sera compared to healthy subjects. There is decreased FRAB, ferric reduction antioxidant potential in diabetic sera. The catalase activity was also found to be reduced. So is the superoxide dismutase activity. Carbonyl content was high in diabetic sera, and MDA content was also high in diabetic sera. So this is a tabular presentation of the antioxidant parameters in normal human sera as well as the diabetic sera. Now direct binding ELISA. Direct binding ELISA, we have found the antigen we used is our modified protein. The antibody is the IgG, is the sera of the diabetic patients. So we have found that the direct binding ELISA we have done with native protein, hydroxyl modified protein, methyl glycoxyl modified protein, as well as hydroxyl and methyl glycoxyl modified protein. And we have found that there is an enhanced binding of the sera samples with the antigen. Highest binding was with the methyl glycoxyl hydroxyl radical modified antigen. So we have done this with 80 sera samples, and we found that in all the cases the sera of diabetic patients bound very strongly, and high absorbance was seen in in diabetic sera samples, which has been attached to methyl glycoxyl hydroxyl radical. So the very high binding sera samples were further selected for inhibition ELISA. Sera number 9, 10, 12, 19 and so on up to 71. There were 12 high binding, 18 high binding sera samples which were selected for inhibition ELISA. So the average absorbance at 410 nanometer in the sera of type 2 diabetes patients binding to native hydroxyl modified, methyl glycoxyl modified and MgOH modified was found to be, in native the absorbance came out to be 0.294. In hydroxyl radical modified the absorbance came out to be 0.436, methyl glycoxyl modified 0.496, and maximum absorbance was seen in Mg glycoxidated protein. Healthy patients, no appreciable binding was observed in healthy patients. Furthermore, inhibition ELISA was done, and in all the inhibition ELISA samples we have found that maximum inhibition was observed in the patients. In the maximum inhibition was seen when the antigen we used is methyl glycoxyl hydroxyl radical modified IgG. The average inhibition is 26.47% in native IgG, 50.6% in hydroxyl radical modified IgG, 50.89% in methyl glycoxyl modified IgG, and 64.84% in MgOH modified IgG. This was followed by isolation of IgG from the sera of diabetic patients, which showed very high percent inhibition, and the direct binding ELISA of isolated IgG was carried out to find out the saturation value, the amount of antigen required for saturation. It was found that the saturation value was least for MgOH modified IgG. Only 40 microgram per ml of IgG is required for saturation. So this is the competition inhibition data of isolated IgG. Again, we have done inhibition with isolated IgG. So all these results pointed out to a conclusion that the structural alteration in IgG upon modification may lead to generation of new epitopes, which is highly immunogenic, and these new epitopes resulted in production of antibodies against the structurally altered proteins. Glycoxidated IgG accumulation and the anti-MGOH immune response may be critical steps in the pathogenesis of diabetes, its progression and severity. MGOH IgG is a potent immunogen presenting unique new epitopes that may be one of the crucial factors in antigen-driven induction of autoantibodies that may serve as potential marker in early diagnosis and prognosis of diabetes. Therapies that control them could potentially improve the quality of life of patients. So this is a pictorial representation of what I wanted to show, that in diabetic patients there is glycoxidation that lead to the generation of new epitopes on IgG. This is an antigen IgG. This is the in vivo situation of what I have explained in vitro. So IgG has neoepitopes due to glycoxidation. These neoepitopes resulted in generation of antibodies in diabetic patients. So when we have collected the serum of diabetic patients, they have high content of antibodies against the neoepitopes, against the IgG. So when in vitro we have modified IgG with hydroxyl, with methyl glycol as well as hydroxyl radicals, so this situation mimics the in vivo situation, as a result of which the antibodies binds highly to this antigen, and so this is detected in our so the result is detected as high binding in direct binding results, inhibition results. So that's all I wanted to present. Thank you.
Thank you very much for this very informative presentation. So, do you have any questions? Yes, please. Microphone first.
Impressive study, actually. Are these results same for type 1 and type 2 diabetes, or is it your work is focused on a specific
my work is on type 2 diabetes, but there have been similar works which have been performed by other students in the university on type 1 diabetes. So in type 1 diabetes also it has been found that there is a very important role of glycoxidation, and so it is same in type 1 diabetes as well. And besides this, numerous other diseases like neurodegenerative diseases, Alzheimer's, Parkinson's also have a role of glycoxidation.
Thank you.
Welcome.
Any other question? Well, if not, then thank you very much, and here is your certificate.